Proteins have very dynamic personality. They are not static like a passport size photo. They have an Instagram photo like characteristic.
Proteins are large molecules containing thousands of atoms. They fold, unfold, and change structure to perform their intended function. Hormones, enzymes, receptors are all proteins. Advanced imaging and microscopy techniques help us visualize three-dimensional protein structures and how they change and interact with other molecules. It can help unravel molecular mechanisms behind digestion, infection, or cancer progression. In the Protein NMR Imaging Laboratory, Professor Ashutosh Kumar uses nuclear magnetic resonance imaging to study the structure and dynamics of proteins.
We are looking at the nucleosome dynamics. So our DNA is packaged inside the cell by this tiny machinery called nucleosome. So we are trying to elucidate how the disorder region in the nucleosome helps in condensing in the form of chromatin.
Ubiquitin is a small protein that marks proteins which need to be disposed of and attaches to them via ubiquitin E3 ligase.
In many of the cancer cases, we know that one of the protein gets dysregulated. The ubiquitin E3 ligase goes and binds to the protein. We try to understand the protein-protein interaction in a ubiquitin E3 ligase called SCF complex, which is implicated in one of the protein get dysregulated in particular kind of cancer, like a prostate cancer there is one, in breast cancer there is another. After understanding this protein-protein interface, can we selectively come up with a peptide-based inhibitor which will intercalate between these interface and breaks the interface selectively so that uncontrolled cell division will not happen and cancer can be checked. So other project we are looking at the metabolic disorder project. So there are two aspects of metabolic disorder project. We use basically the metabolites which is coming from serum to early predict if somebody is going to have the diabetes or so. We are taking the serum from different person, recording the metabolites using NMR and mass spec, and then applying AIML tool predict early what kind of dysregulation in a person can happen. And then we are again understanding how the peptide based drugs interact with the receptor to produce the downstream cascading signal, and how we can modify or smartly design the drug that has a better efficacy and less side effect.
Professor Prasenjit Bhaumik's protein crystallography laboratory explores and describes the structure-function relationship of proteins and structure-based design of enzymes and drugs.
When a protein functions, it might undergo different conformations. Let's say this is a protein structure, but this can do multiple different functions. Now I think I have to hold a pen. I cannot hold a pen like this. So for holding a pen, my fingers have to bend, and eventually then it can hold a pen. So that means the protein has to change its conformation for holding a particular molecule. So that's a different conformation. There's possibility a protein will have multiple different conformations for different functions.
Professor Bhaumik studies the structure of glutamate dehydrogenase, or GDH, an enzyme that plays an important role in amino acid metabolism. The team aims to decipher how the structure of GDH helps it to act as a catalyst and to understand how this enzyme recognizes its helper molecules.
So here you can see this is the enzyme glutamate dehydrogenase. In the native state, it's a hexamer. It has 6 molecules, one monomer. You can see the structure is monomer. It has 2 domains, but eventually 6 molecules come together to form a beautiful hexamer, and the actually active enzyme is a hexamer. What you can see here, the 6 molecules are together.
Professor Bhaumik's lab also studies plasmepsins, a group of enzymes produced by the malarial parasite.
The structures are very important to understand the binding of inhibitors. We have determined multiple crystal structure of plasmepsins, and based on the structures, we have developed compounds. Those can inhibit only the parasite enzyme, not the human enzyme. And here you can see the multiple crystal structure of those proteases, which are plasminogens complexed with different inhibitors. And based on our biochemical studies, we feel that those are the compounds which can be developed into an antimalarial drugs.
Professor Sandip Kaledhonkar uses cryo-electron microscopy to study structures of large protein complexes and other biomolecules. Biomolecules are frozen to very low temperatures, below -150 degrees Celsius, to observe their structures. Cryo-EM is one of the best ways to visualize large biomolecules, as it has low radiation damage and crystallizing the protein is not needed. Professor Kaledhonkar's lab also observes dynamics of biomolecules using single particle cryo-EM.
When it comes to the cellular level, we can have the cryo-electron tomography technique, where we can get the sections of the cell and we can look into the organelles of particular organism. Many times we want to understand how the particular cell behaves under certain conditions. Let's say it could be in terms of cancer, stress condition, or you have some pathogen, how this pathogen is interacting with the host cell.
Professor Kaledhonkar's lab studies ribosome complexes from tuberculosis and malaria causing pathogens. Ribosomes are the targets of many clinical drugs. The team is trying to understand the basic principles of how the mutations that make the pathogen drug resistant happen. Professor Kaledhonkar also studies the structure of phages, or viruses that attack bacteria. The team is studying phages that attack tuberculosis causing bacteria.
In the phage interaction with the mycobacterium, we want to learn what are the partners over here and how exactly phage is infecting the mycobacterium, and that we want to capture through single particle cryo-electron microscopy as well as cryo-electron tomography.
Advanced imaging techniques open up several possibilities for studying structures of proteins and offer valuable insights into interactions between biomolecules. With the nuclear magnetic resonance imaging, crystallography, and cryo-electron microscopy facilities and expertise at IIT Bombay, researchers are set to unravel new biomolecular secrets.